Hello, everyone. Our lesson for today is about microbial cell wall and intracellular structures. This lesson focuses on the bacterial cell wall. Specifically, it will make you understand how the cell wall functions, what is its composition, what are the basic types of the cell wall, and how it relates to microbial classification. Moreover, the structures and functions of the possible components that can be found inside the cell of bacterial cells and some cellular processes that occur inside the cell are also discussed in this lesson. So at the end of this lesson, you are expected to describe the function of the cell wall, identify the composition of the cell wall, differentiate gram-positive from gram-negative bacterial cell wall, relate the gram-stain mechanism to the structure of the cell wall, identify the components found inside the bacterial cell wall, and describe the structures and functions of the composition of the components found inside the bacterial cell wall. Before we begin our discussion, let me ask you this question. Does the composition of the cell wall determine its functions? What do microorganisms use for their various metabolic processes? I hope you will be able to answer that one as we end our discussion for today. So the cell wall of the bacterial cell is a complex, semi-rigid structure responsible for the shape of the cell. So if you can see here, this is the electron micrograph of a representative bacterial cell, and being pointed here, this arrow is the cell wall, no, of these bacteria. So the cell wall surrounds the underlying fragile plasma or cytoplasmic membrane and protects it from no, the and protects it in the interior of the cell from adverse changes in the outside environment. And almost all prokaryotes have cell walls, so the bacteria as well as the archaea, they have their cell walls. And even some groups of eukaryotic organisms now have cell wall, like the fungi. So what are the functions now of the cell wall? So the cell wall prevent bacterial cells from rupturing when the water pressure inside the cell is greater than that of the outside the cell. And it helps maintain the shape of a bacterium and serves as a point of anchorage for the flagella. So as the volume of a bacterial cell increases, its plasma membrane and cell wall is extend as needed. Clinically, the cell wall is important because it contributes to the ability of some species to cause disease, and it is the site of action, no, of some of the antibiotics. So at the end, no, or the last module, we'll talk about um, uh, control of growth of microorganisms, and we will be talking about actions, no, of some antibiotics. So the chemical composition also of the cell wall is used to differentiate major types of bacteria, which we will talk about later on. So what is the composition of the cell wall? What are those that comprises the cell wall? So the bacterial cell wall is composed of a macromolecular network called peptidoglycan, which is also known as myelin. This is present either alone or in combination with other substances. So the peptidoglycans consist of a repeating disaccharide attached by polypeptides to form a lattice, no, like a net, that surrounds and protects the entire cell. So the disaccharide portion is made up of monosaccharides called the NOG and the NAM. So NOG stands for N-acetylglucosamine, while NAM stands for N-acetylmuramic acid. Okay, so these are actually glucose derivatives. So this is the chemical structure of both of these disaccharide. So disaccharide because this is a monosaccharide and another monosaccharide. So they are joined together by this glycosidic bond. So it is actually a disaccharide, but it is a glucose derivative. Okay. So together, the carbohydrate backbone, so the NAG and the NAM, forms the glycan portion. However, no, connecting this glycan portion, the repeating disaccharide, are the tetrapeptide side chains. So this makes up the peptide portion. So they make up the peptidoglycan. So that means a combination of a peptide, a tetrapeptide, and a glycan portion. So we already know the glycan portion that is made up of disaccharide, the NUG, and the NUM. But how about the tetrapeptide side chains? So it is a tetrapeptide side chains, meaning there are four peptide bonds found. However, there are only four amino acids present because the last peptide bond is formed between the L-alanine and this functional group of the N-acetylmuramic acid. So together with alanine, commonly found in some or in most bacterial cell wall are glutamic acid, diaminopimelic acid, and D-alanine. Although the composition may differ in some species of bacteria. Now, there are two types of cell walls in bacteria. So, a cell wall could be gram-positive or they could be gram-negative. How do they differ from each other? A gram-positive cell wall, okay, has a thick layer of peptidoglycan, okay, many layers. So, they form a thick and a rigid structure. Also, gram-positive cell wall contain tachyoic acids. So, two types of tachyoic acids are found in gram-positive cell wall, the lipotachyoic acid and the wall tachyoic acid. Wall tachyoic acids are found embedded within the peptidoglycan layer, while lipotachyoic acids are those that span from the peptidoglycan layer and is embedded towards the cell membrane of the gram-positive bacteria. The gram-negative cell wall, on the other hand, contains only a thin layer of peptidoglycan. No, but what makes it different from gram-positive, aside from having a thin layer of peptidoglycan, is the presence of the outer membrane. So if you notice here, aside from the inner membrane or the plasma membrane of the gram-negative bacteria or bacterial cell, it also has an outer membrane. So the peptidoglycan is like a filling, no, between these sandwiches or between these lobes. So parang they make up a sandwich with a peptidoglycan filling. So actually the outer membrane together the peptidoglycan makes up the cell wall of this gram-negative bacterial cell. Now the gram-negative bacterial cell wall has outer membrane with LPS now that are embedded in the phospholipid bilayer. So the LPS or the lipopolysaccharide is composed of the lipid A, core polysaccharide, and O polysaccharide. So peptidoglycan here in gram-negative bacterial cell is bonded to lipoproteins. So this is the lipoprotein. So the lipids are covalently linked to proteins. So lipoprotein, a combination of a lipid and a protein, found in the outer membrane and is in the periplasm. The periplasm is the gel-like fluid between the outer and the inner membrane or the plasma membrane. Okay, so this gel-like fluid here, where the peptidoglycan layer is embedded, is the periplasm. But what connects the peptidoglycan to the outer membrane are structures called, or are molecules called lipoprotein. And you have here the lipopolysaccharide component. Now, the outer membrane of gram-negative cell wall, as we have said, is composed of lipopolysaccharides, lipoproteins, and phospholipids. And this outer membrane has specialized functions. The strong negative charge is an important factor in evading phagocytosis, no, being eaten by other cells, and the actions of complements. So complements are the those that are released by cytotoxic T cell to kill those cells that are bounded with antibodies. No, so in order to evade that, the gram-negative cell wall has outer membrane that could help the cell survive. No, the action of complement. The outer membrane also provides a barrier to certain antibiotics like penicillin. So penicillin actually targets the peptidoglycan layer. So with a barrier, the penicillin could not really penetrate into the peptidoglycan layer of gram-negative bacterial cell. Aside from that, it could also be a barrier for digestive enzymes such as lysozyme, detergents, heavy metals, bile salts, and certain dyes. So as what you've said, lipopolysaccharides are found in the outer membrane. It is a large complex molecule that contains lipids and carbohydrates and consists of three components, the lipid A, corpolysaccharide, and O polysaccharide. The lipid A is the lipid portion of lipopolysaccharide and is embedded in the top layer of the outer membrane. So when a gram-negative bacteria die, they will release lipid A, which functions as an endotoxin. Lipid A is responsible for the symptoms associated with infections by gram-negative bacteria such as fever, dilation of blood vessels, shock, and blood clotting. The core polysaccharide, on the other hand, is attached to lipid A and contains unusual sugars. The role is structural, so they provide stability for the outer membrane. Lastly, the O polysaccharide is the one that extends outward from the core polysaccharide and is composed of sugar molecules. So the O polysaccharide functions as an antigen, the one that elicits immune response in the cell, and is useful for distinguishing species of gram-negative bacteria. Now, these bacteria, because of the differences in bacterial cell wall, a technique was developed by Hans Christian Grams, to in order for us to determine which type of cell wall the certain bacteria uh, has. It's very important because in early diagnosis, especially in medical cases when infection has already gone severe, so the first thing to do is to determine the gram the gram reaction or the cell wall of the bacteria in order to administer the proper antibiotics, and also in classification, so gram staining is needed. So this is the steps on how you would determine the cell wall composition of bacteria, if the bacterial cell has gram negative or gram positive cell wall. So the mechanism is based on differences in the structure of the cell walls of gram positive and gram negative bacteria and how they react to the various reagents. So the reagents are crystal violet as the primary stain, mordant, the iodine as the mordant, then decolorizer, alcohol as the decolorizer, and safranin as the counter stain. So what they do first is you will also be doing this in your laboratory actually. So what you'll do first is you stain the cells with crystal violet. So the crystal violet will bind directly to the peptidoglycan layer of the bacterial cell wall. Then you add an iodine as the mordant. So what iodine does is inside in the peptidoglycan layer, the iodine will bind with crystal violet, so they form a crystal structure. So that when you wash the stain, it will not be washed out because they are trapped inside the peptidoglycan layer because they had formed a crystal structure. Now, an alcohol is applied as a decolorizer. So the decolorizer, the alcohol will degrade the outer membrane of the gram-negative bacteria. Now, so if crystal violet and iodine has already formed a lattice around here and they cannot be washed out because of the thin layer of the peptidoglycan layer, and because of this, but when the decolorizer is applied, it will degrade the outer membrane and some of the peptidoglycan layer so that the crystal violet and the iodine is also washed out. While in gram-positive bacteria, it will not be washed out because there is a thick layer of peptidoglycan. So what happens is that during crystal violet staining, both gram-positive and gram-negative cell will still be colored violet or blue. After the application of mordant, both of the cells will be colored blue or violet. However, after decolorizer, the gram-negative bacterial cell will be colorless, while the gram-positive bacterial cell will remain blue. And last, after decolorizing, safranin is added as a counterstain. Okay, so what happens if you add safranin to a gram-negative cell? Of course, since a gram-negative cell has already colorless, it will form no pink to red color. It will just retain the saffron in color, which is red. So it could be, depending on the application, pink to red color. However, since the gram-positive cell has still crystal violet in its cell wall, the application of a red color will make it a more violet darker. So it could be violet to blue. So after your gram staining reaction, you will notice if it is pink to red, that means the cell is gram negative. If it is blue to violet, then that the bacterial cell is a gram positive bacteria. So here's a result of one of my thesis students who has investigated antibiotic resistant bacteria in swine. And this is his comparison of a gram reaction. You have here an E. coli, which is a gram negative bacteria, and Staphylococcus vitellinus, which is a gram positive bacterial cell. Okay. So this is a transmission electron micrograph of a bacterial cell that shows the different structures that are found inside the bacterial cell. So they have plasma membrane that is underneath the bacterial cell wall. So we will be talking about all the components here. So last time we talked about those that are found external. Then we also talk about the cell wall itself, and now those that are found internal to the cell wall. So yeah, they had the bacterial cell have the plasma membrane, the cytoplasm, the nucleoid, ribosomes, inclusions, and endospores. So the plasma or the cytoplasmic membrane or the inner membrane in gram-negative bacteria is a thin structure lying inside the cell wall and enclosing the cytoplasm of the cell. In prokaryotes, it consists primarily of phospholipids, which are the most abundant chemicals in the membrane, as well as proteins. So you have here phospholipid molecule. So it is composed of a hydrophilic head and a hydrophobic tail. No? So they make up a double layer because the hydrophobic tails forms hydrophobic interaction. No? So that all the hydrophilic heads are in contact with the watery environment outside and inside the cell. So the plasma membranes serve as a selective barrier through which materials enter and exit the cell. Plasma membranes have selective permeability, so sometimes called semi-permeability, which means that certain molecules and ions pass through the membrane. Others are prevented from passing through it. So what determines if a certain molecular ion can pass through it? So it depends on the size, charge, and polarity of the molecule. Plasma membranes are also important for the breakdown of nutrients and the production of energy. So in the membrane, some are found, or in some species are enzymes that help in energy generation in the form of ATP. Others have components that are able them to harvest energy from the sun or other sources in order to produce their own food. So there are membranous structures in some cells that are called chromatophores or thylakoids that harvest energy to photosynthesize. So how do materials move in and out of the bacterial cell? So materials move across plasma membranes of both prokaryotic and eukaryotic cells by two kinds of processes, passive and active. So in passive processes, substance cross the membrane from an area of high concentration to an area of low concentration. So this is what we call concentration gradient or difference without the use of energy. Okay. However, in active processes, this is a movement against a concentration gradient. It is not go with the flow, but you are going against the flow. So you need not to spend energy, just like you when you are swimming in the river, in the ocean. If you just go with the flow, you don't really spend a lot of energy. But if you swim against the flow of water, you really need a lot of energy. Okay, so movement of substances across the membrane is also like that. So passive processes, include simple diffusion, facilitated diffusion, and osmosis. So if it is just the movement of certain substances across the membrane from area of high to low concentration, it's just simply diffusion. But we know that there are permeability factors like polarity, size, and the charge of the molecules. If it is, even though they are high concentration on the other side and low concentration on the other side, if no they cannot pass through because they are very big or they are polar, then they could use other uh, gate. So with this, there are what we call transporter proteins. So they could be non-specific or specific transporters that would help him move across the membrane. So just like this is a specific transporter of glucose. So glucose cannot pass through this because it's a big molecule. So what it does, it uses a transporter to pass through. So this is what we call facilitated diffusion. No, so a transporter protein helps in the transport of the molecules from an area of high concentration to a low concentration. Still, there is no energy that is being used. Now, there are also what we call channels, no, or canals within the cell. We know that water is highly polar, so they cannot really pass through. They are in theory, they are not really allowed to pass through a non-polar environment. However, they are very small, so some of them, and since the hydrophobic interaction is just a weak interaction, so some of them could pass through the membrane. However, most of the water will enter the cell or go out of the cell via these water channels called aquaporins. Now, because of that, no, there in many cases there is a net movement of solvent molecules across a selectively permeable membrane from an area of high concentration of solvent molecules. That means there is low concentration of solute molecules because the concentration of solvent and solute are inversely proportional to each other. So we call this net movement of solvent as the osmosis. The diffusion is the movement of the solute, but osmosis is the diffusion of water. So with that, we refer to different tonicities. Now that we are referring to, we identify three tonicities that refers to the concentration of solute outside the cell. So isotonic, hypotonic, hypertonic solution. So in an isotonic solution, there is equal concentration of solute inside the cell. Okay, so what happens is that since they're equal, so the number of water, the concentration of water that is going out and the water that is going in is equal. So isotonic. There are also hypotonic solution, meaning there is low concentration of solute outside and high concentration of solute inside. So if there is low concentration of solute outside, there is high concentration of solvent outside the cell. And since there is low concentration, and since there is low concentration of solute inside the cells, what happens is that although there are water that comes out and there are water that comes in, more of the water is coming inside. So what will happen? Now, just like a balloon that na napasobraan og butang og daghan nga air, what will happen? It will burst. So just like a cell, if there is so much water that is coming in, it will eventually lies. However, if the cell is in hypertonic solution, that means there is higher concentration of solute outside than inside. That means there is lower water concentration outside, so more of the water is going out, and so the cell membrane, the cytoplasm is lies. So this is what we call plasmolysis. This is actually the principle. When you have some people have their sore throat or bacterial infection, what they do is they gargle salt water, or they gurgle a salt solution. So that is to provide a hypertonic environment for the bacterial cell to lyse. Although in some it may be effective, but in others pwede po dili. So a bacterial cell needs to be always in isotonic solution with the environment so that it will survive. So it really needs all the mechanism that it could use in order to stay alive. in different environmental conditions. Now, if there is already expenditure of energy, that is already called an active process. So when a bacterial cell is an environment in which nutrients are in low concentration, the cell must use active processes such as active transport and group translocation to accumulate the needed substances. Okay, so the cell uses energy in the form of ATP to move substances across the plasma membrane. Among the substances that are actively transported are ions, for example, sodium, potassium, protons, calcium, and chloride, amino acids, as well as simple sugars. Now, active transport could also be in the form of could also use ion gradient to co-transport some molecules. So we have here co-transport of a substance via these ions. So if there is ion gradient that occurs, they could be transported together with the target molecule, or they could be transported in a different direction. This is what we call antiport, and this is symport. Now, a uniport could be active or passive depending on if there is energy expenditure. Then a uniporter is an active process. Okay, so that was all for that were all for the plasma membrane. Now let us talk about the cytoplasm. The cytoplasm for a prokaryotic cell. Now the term refers to the substance of the cell inside the plasma membrane. It is about 80% water and contains primarily proteins, carbohydrates, lipids, inorganic ions, and many low molecular weight compounds. Inorganic ions are also present in much higher concentration in cytoplasm than in most media. Cytoplasm is thick, aqueous, semi-transparent, and elastic. The major structures in the cytoplasm prokaryotes are nucleoid containing DNA, particles called ribosomes, and reserve deposits called inclusions. So protein filaments in the cytoplasm are most likely responsible for the rod and helical shapes of bacteria. Prokaryotic cytoplasm lacks certain features of eukaryotic cytoplasm, such as cytoskeleton and cytoplasmic streaming. The nucleoid is another structure that you can find inside the bacterial cell. It is usually containing a single long, continuous, and frequently circular arranged thread of double-stranded DNA called the bacterial chromosome. Although in some species they could be linear, no chromosome, and they could be occur in multiple copies, not just one. But most of the bacterial cell have single, circularly arranged double-stranded DNA. Now, aside from that, the nucleoid also you will find plasmids. Okay, so these are extra chromosomal genetic elements that are not connected to the main bacterial chromosome. If this is the bacterial chromosome, then some of the genetic materials, extra chromosomal genetic materials, are called plasmids. These are the ones that are acquired by the bacteria from their environment. So some confer selective advantage like antibiotic resistance or heat resistance or heavy metal resistance, the ones that help them survive in a certain environment. It could be inherited by the other cells, but some of these may be lost in the future generations of the bacteria. So usually these are taken through horizontal gene transfer like conjugation. Ribosomes are found in the nucleoid and is associated with the bacterial chromosome. They are the ones that help in protein expression or in the protein synthesis or in the translation. It is made up of small subunit and large subunit. So 30 Svedberg and 50 Svedberg, and altogether they make up 70 Svedberg ribosome. Inclusions, on the other hand, are reserve deposits. So in certain conditions, when they are plentiful, so nutrients that are needed by the bacterial cell could be accumulated inside and use them when the environment is already deficient. So that it is a form of saving nutrients in order to save for the tough times. So these are some examples of inclusions that you can find in some types of bacteria. Now, finally, found inside in the bacterial cell wall are endospores. So when essential nutrients are depleted, so certain gram-positive bacteria, such as those of the genera Clostridium and Bacillus, will form specialized resting cells called endospores. Okay, so I think we have already talked about this before. So the process of endospore formation is called sporulation. So what happens is that if the bacteria senses that they could not probably survive this condition, what they do, they replicate their genetic material. They enclose it in a very tough covering. Okay, very tough layer, and then it forms a spore coat, and then it is freed from the cell. Now this in the spore will become dormant for many years until a condition is that is favorable for their growth is um no is encountered, and so they will germinate. So many of these endospores will last for many years. So here are summary questions to help you review our lessons now for the day. Why is cell wall important in a microbial cell? What are the components of the microbial cell wall? How do gram-positive cell walls differ from gram-negative cell walls? What is the role of gram staining in microbial identification? What are the different components found inside the bacterial cell wall? And what are the structures and functions of these components? So I hope you will be able to answer this one to help you to fully understand the topics that we have discussed today. Okay, so before we end, I would like to acknowledge Luis Maria Shardita Esrosal for helping me prepare the slide presentation for this topic. You can contact us, no? Here's my email, and you can contact your lecturers for any questions or clarifications regarding this lesson. You can also key in your questions in our Facebook Messenger group chat. So thank you very much, and see you again in our next lesson.